All right, welcome back. We're on part 2 of chapter 6. So now we're going to talk about some functions that are specific to skeletal muscle. Um, no, yeah, that's what I'm going to say for now. There are other cells that do have some of these same functions, but these are very special to skeletal muscle as well, okay? So the first is irritability. What that means is they are able to take in a stimulus and respond to that stimulus. Then they can contract. So contraction is just the shortening and then going back to its normal length would be extensibility. And then resume resting or length after extending would be the elasticity of skeletal muscle, okay? So if a skeletal muscle is going to respond to a stimulus, then there has to be some type of a stimulus that it can respond to, and that's a nerve. So a neuron is a cell that sends signals or takes in signals, information. And so in this case, a neuron can send a signal to skeletal muscle, and then that skeletal muscle will respond by contracting. When a neuron does this, it can actually send signals to multiple cells within a muscle, but only one neuron can stimulate any given muscle fiber. So you can have one neuron innervating multiple muscle fibers, but only one neuron can innervate any given muscle fiber. I hope that makes sense. And actually, I think the next picture will show you. So here we have two different types of neurons. So we have a purple and then the red, right? So this one is innervating this muscle fiber and this muscle fiber, so it can innervate more than one. And then the purple one innervates this one, and this one, so it's innervating more than one. But you don't have purple and red innervating the same muscle fiber. That's what I'm trying to say, okay? So let's look at, oh, and this is another picture showing the neurons innervating different muscle fibers, okay? So what now I'm going to talk about is what happens when a neuron does send a signal to innervate a muscle fiber. And so this would be at a region of the muscle called the neuromuscular junction. This is where a neuron and the muscle fiber, the sarcolemma of the muscle, actually come together. So here is the end bulb or axon end bulb of a muscle fiber, or I'm sorry, axon end bulb of a neuron. And then this is the sarcolemma of a muscle fiber. In between, if you notice, there's actually a gap between the two, the neuron axon end bulb and the sarcolemma. That gap is called the synaptic cleft or synapse. And this is where the neuron will release chemical messengers called neurotransmitters to stimulate the neighboring cell. So they don't actually touch. They actually release chemicals that move across this gap and then stimulate the next cell. In the case of skeletal muscle, the neurotransmitter that's in these little tiny vesicles here is called acetylcholine or ACH. So acetylcholine right here is typically written as ACH for short, okay? So what happens is a stimulation comes along down, runs down the axon to the axon end bulb. And when it does this, my dog is like being weird. You see her back there, right? Right. Oh, whoa, what just happened? Hold on. Okay, hold up. That was weird. I don't know. It seemed like all of a sudden my screen, my face just disappeared, and I was like, wait, what's going on? So I didn't know. Anyways, my dog was sitting back there, but now she's gone. She walked off. So what's happening is once that message moves down the axon and hits that end bulb, it stimulates calcium gated channels in the end bulb to open up. and calcium rushes in. Calcium then binds with the vesicles, allowing the vesicles to release their chemicals via exocytosis. So remember that from our previous chapter, we talked about exocytosis. The vesicle binds to the plasma membrane of the axon end bulb and opens up, releasing the neurotransmitter, and in this case, it's acetylcholine. So acetylcholine then moves across the synaptic gap or cleft and binds to receptors on the sarcolemma. Binding of those receptors opens up sodium gated channels. So sodium is able to move in, and sodium rushes in, causing depolarization of the muscle fiber or a stimulation of the muscle fiber. So that triggers more sodium gated channels to open up, and then potassium channels open and potassium rushes out. So before, so I'm going to get a little bit more into it in a minute, but once sodium and potassium have exchanged places, we have sodium-potassium pumps that are going to pump the sodium against its gradient out of the cell, and potassium is going to be pumped back into the cell using energy of ATP. And then we have these enzymes that break down acetylcholine. As soon as acetylcholine binds, these enzymes called acetylcholinesterase, or ACHE for short, will start to break down acetylcholine. So we're releasing the acetylcholine into this gap. The acetylcholine binds, opening up sodium channels, and then is broken down immediately. And so we have to keep having more and more acetylcholine releasing so that we can keep that muscle contracting. All right, so what we're going to do now is we're going to look at each of these steps a little bit closer. So the first thing that happens is we have that nerve impulse reach the axon terminal, or this axon end bulb, okay? So here you see that happening. Step 2, the calcium channels open and calcium enters into that end bulb. What happens then is the calcium will bind to the vesicles, and that allows the vesicles to release their materials, their acetylcholine, via exocytosis. Let's see if this works. Step 3, you have calcium enters, and that allows the calcium to bind to the vesicles, and the vesicles release their chemicals via exocytosis. The chemical, again, is acetylcholine. Once acetylcholine is released, it diffuses across that synaptic cleft or that gap, and it binds to the receptors on the sarcolemma, which then allows those protein channels to open, allowing sodium to rush in. Okay? So as sodium channels open, sodium rushes in rapidly. Sodium moves down its concentration gradient from the outside of the cell into the cell, changing the inside of the cell to a more positive charge. Potassium moves much more slowly, but moves out of the cell. And so, but it's also moving down its concentration gradient from in the cell, which is a higher gradient, than to out of the cell. So you end up having depolarization inside the cell because we get a lot of positive charges moving in compared to what's leaving the cell. Nope. And as all of this is happening, acetylcholine esterase enzymes are breaking acetylcholine down. So as soon as they bind, acetylcholine esterase breaks it down. And another one has to bind, and another breaks it down, and another binds, and another breaks it down. So this is continuously happening until we no longer need that muscle contraction, and then no more acetylcholine will be released, and the muscle will relax. And so this is that same picture that we were looking at before. I'm moving my head so you can kind of see everything. I'm going to stop the video here, and hopefully the next video will be a little smoother because I don't, this was weird. It wouldn't, I couldn't move my slides the way I'm supposed to. So I will stop it here and try to get a better video for the next section. Bye.